Welcome to the third episode of the Environmental Biotechnology Innovation Centre podcast, EngBio Tech. Today we'll be exploring the use of engineering biology in combating oil pollution. As you may know, as well as our podcast, we've recently released the first of our webinar series, Engineering Biology in Action. If you head over to our website, ebicentre.co.uk, you can watch as our EBIC experts discuss microbial engineering, toolsets and genetic engineering methods. It's well worth a watch. But today for the podcast, joining me are Dr. Boyd McKew of EBIC and the University of Essex, and Senior Research Officer Dr. Hannah Adams from Boyd's team at Essex, and of course part of the EBIC team as well. So thank you very much, both of you, for joining us. It's great to have you with us. Boyd, if you wouldn't mind starting things off, just let us know a bit about yourself and your background.
Yeah, thanks, Nicola, and thanks for inviting us today. My background is I'm actually currently a senior lecturer at the University of Essex. I actually came here to study a marine biology degree, what seems like a very long time ago now, back in around 2000. And so I've been here for 25 years, but I actually did my PhD here. As well as an undergraduate degree, which was actually when I met Fred, who's the leader of EBIC.
Yeah,
we worked on a project together that time ago when Fred was a postdoc and I was a PhD student on marine oil pollution, which was the topic of my PhD. And since then I've been a postdoctoral researcher, and then some years back, I got independent fellowship in synthetic biology to look at oil biodegradation pathways in bacteria, and moved into a lectureship, a senior lectureship. And research-wise, I'm a marine biologist, but I specialise in molecular microbial ecology and physiology. So I do a lot of work with marine pollution and looking at biodegradation of different compounds, particularly oil and hydrocarbons. But I do have a range of other marine projects as well, looking at marine biodiversity in different systems across the world, actually.
Oh, that's fantastic. That sounds really interesting. I didn't realise that you and Fred had known each other so long as well.
Yeah, very long time.
Excellent. Oh, that's great. Thank you so much. Hannah, I would love to hear more about you as well. And do you mind letting us know a bit about yourself and your work as well?
Yes, of course, Nicholas. So this is actually my first postdoctoral position, working with Boyd. So my previous work, when I was doing my PhD studies, was in protein structure and function relationships, primarily looking at metalloproteins. Mm-hmm. And those are involved in the production of nitrous oxide, so that's sort of an environmental side of things. I specialised in using techniques such as X-ray crystallography and lots of different types of spectroscopy, activity assays and kinetic studies.
Oh, fantastic.
So my previous work was very different to what I'm doing now. So the stuff I'm doing with EBIC and oil pollution is all quite new to me, so I'm learning a lot through Boyd. And it's all quite exciting.
Oh, excellent. That's really good. So what was it? What drew you to this postdoc position then, if this was your first one? Is it sort of to try something new and to kind of get involved with the new projects?
Yeah, so I was really interested in the environmental side of things. As I said, I did a little bit during my PhD work. And the work that Boyd had been doing previously just really interested me, looking at oil pollution and finding new ways to tackle that with engineering biology.
Fantastic. That sounds great. Thank you. So to start us off, could you explain a bit about some of the major environmental and ecological consequences of oil spills? Maybe things that the public don't fully appreciate or aren't aware of?
I think probably most people are more aware of like the immediate effects of oil spills. So when you think of oil spills, you think of like the news stories where you've got birds and other marine life all coated in oil and stuff, and those sort of very immediate effects of it. But there are also much longer lasting effects, which probably people are not quite so aware of.
Yeah, I think I'd agree with that, to be fair, because if even me personally, I think of it, you think of like the oiled birds and then the people sort of trying to wash them off with like fairy liquid and things like that. So that does make sense.
Yeah, what's quite interesting as well is that you know, throughout all the oil spills, the major oil spills that we've had in history, it's fair to say that no oil spill is ever the same. So the effects can be really different depending on type of oil that's spilled, the scale of the spill, but also the particular environment it's in, because they do happen all around the world, and you know, spills in the tropics are very different to what we see in temperate systems and also the Arctic. But yeah, one thing that perhaps, as Hannah was alluding to, that is not kind of realised, how long it can sometimes take some systems to recover. So if we think about the Exxon Valdez spill, which was back in 1989, for example,
yeah,
it doesn't even rank as one of the biggest spills. It's probably about 35th on the list in terms of scale, but it was in a pristine environment in the Arctic. So many organisms were affected by oil, you know, including birds, otters, but a whole host of shellfish, for example. But, you know, when people were looking in the following years, the spills disappeared from the news, but the data actually showed that even 25 years after the spill, there were a number of species that had still not recovered from the effects of that oil spill in terms of their population size.
Oh wow.
And you know, also the scale of the amount of different types of organisms that are going to be affected, because we once worked on an oil spill in Galicia, it was the Prestige spill, around about the time of my PhD that was. But you know, some other researchers at the time are measuring the effect on some of the contaminated beaches, and you could see as much as sort of 66% of the total species richness actually disappearing from the coast. And you know, historically we've had some major spills around the UK and Europe as well, and the Brer spill that was up in the Shetlands, for example, you know, had knock-on consequences that some of the shellfish fisheries there were sort of shut down for periods of up to 7 years. And there was another spill that goes right back to the 1970s, so well before my time at university. But it was something that we got involved with really only in the last couple of years, because perhaps people don't realise that when oil gets into sediments, you know, particularly when it starts to get buried in sediments and it goes into layers where there's a lack of oxygen, the biodegradation can be exceptionally slow. And there was an oil spill not far from here in Lowestoft where, as part of the remediation, a lot of oil was actually, the decision was made to bury it off the beach. And because it's a naturally retreating coast, over the last 50 years, the sea has been eroding it. A storm called the Beast from the East actually, as it was eroding the sort of dune sea wall away, unearthed the kind of graveyard of 50-year-old partially degraded oil onto the beach. So, you know, that's seeing effect some 50 years later.
Yeah, so these things can be over decades then, basically. Yeah, thank you so much. So we're talking about the oil spills and things like that. Are they the main sources for oil pollution in the environment? I think to most people they sort of seem to be the most obvious to us, but is that actually the case?
It's not actually, no. It just tends to be that they're the ones that the public certainly hear the most about, because you know, it tends to be can be quite catastrophic sometimes. It's also a major news event when an oil tanker, for example, runs aground, or where there's, you know, a big pipeline spill, or even such as the Deepwater Horizon, which was a wellhead blowout, you know, from the extraction from the environment. But actually, kind of tankers and platforms, whilst being the most highly covered, are by no means one of the biggest sources of oil actually into the sea. It's always been quite difficult to estimate that. But, you know, when you look at some of the data about where oil and hydrocarbons actually come from, it's kind of worth remembering that, you know, oil is a natural product. It's formed over geological timescales in marine sediments. So there's probably 10 times more oil that actually gets into the sea from natural oil seeps at the sea surface that come from these types of spills. But even if we take anthropogenic kind of effects, by far the biggest source is the runoff of oil and, you know, oil-based products containing hydrocarbons that come from land, that basically run off land into the sea or into the rivers and into the sea, which is at least 100 times greater than the total amount that comes from these large oil spills that we hear about on the news. And
oh wow,
kind of in the region of 1.2 trillion tons per year globally.
Yeah, there's also a lot of natural sources of hydrocarbons that enter the sea as well. So from like plants, algae, and cyanobacteria, which are a type of photosynthetic bacteria. Some forms of cyanobacteria are estimated to input alkanes into the sea that could be like 100 to 500 times fold greater than all the crude oil sources combined. So that is a lot going in.
That's amazing. I had no idea about that.
Yeah, and it's kind of really interesting. But then, you know, when you consider all this, that you've got this kind of constant low-level hydrocarbons entering the marine systems from many different sources, it's hardly surprising that, you know, evolution has selected for specialist hydrocarbon degrading bacteria that can degrade a very wide range of hydrocarbons, which are the main chemical constituents of crude oils globally. And you know, the specialist bacteria that I've been working with for many years are ubiquitous across the oceans. And, you know, we find them spanning the polar regions to the tropics, and right from the sea surface to the very deepest parts of the ocean, you will find hydrocarbon degrading bacteria, and many different types that have got different genetic capabilities to degrade the many hundreds to thousands of different hydrocarbon constituents that you can find within crude oils.
Oh wow, that's fantastic. So how effective are current oil spill cleanup strategies? Things like chemical dispersants, mechanical removal, bioremediation, which we've touched on a little bit. How can the use of engineering biology improve oil spill response? Does that have a role to play?
Well, it does have a role to play, definitely. So obviously you mentioned initially some of the current oil spill cleanup strategies. So there are many effective strategies, which mostly can include containing oil. They use booms to contain it, and you can have special skimmers that can help clean up. But these can be quite challenging things to use in very large oil spills. Other methods that we have is including in situ burning, but that can't be done anywhere near land. That can only be done on spills that are far out in the ocean, so it's not going to have an effect on anything else.
Of course, yeah.
Yeah, and you kind of mentioned chemical dispersants. I mean, they've actually kind of been an invaluable response tool for decades, really, within kind of oil spill industry. But it's worth kind of mentioning that, you know, to use a dispersant is actually considered as a bioremediation strategy, because if we don't remove oil from the environment, the primary loss route is biodegradation by natural marine bacteria. And the reason that decisions are made to actually disperse oil, it's usually obviously to avoid oil reaching land if models predict that, you know, an oil spill is going to move landward, where we know it causes significant damage when it hits coasts. But if you can disperse an oil slick actually into the sea, the benefit of dispersants which contain surfactant components, chemical surfactants, essentially break up the oil slick in tiny droplets. You know, the oil sits on the surface because of difference in density, forms really large slicks and spreads to very thin layers. But when it's dispersed, it will break it up into tiny droplets. And those tiny droplets actually make the oil and the hydrocarbons much more bioavailable to oil degrading bacteria that naturally live in the sea. it has the effect of increasing the surface area to volume ratio, and it allows oil degrading bacteria to actually attract to these small droplets, make them bioavailable and take them into the cell. And they use the oil as both the carbon and the energy source for growth, and so their populations can actually explode. And they'll completely mineralize an oil hydrocarbons, leaving byproducts of just carbon dioxide and water, for example. But another important reason for doing it is that, if you imagine oils on the surface, it's actually in a very high concentration, and bacteria require other resources. They can get carbon from oil because, hydrocarbons obviously just contain carbon and hydrogen, but bacteria need other resources such as nitrogen and phosphorus to make amino acids and DNA and RNA to actually drive cellular growth. What will happen when you've got a huge increase of carbon in the form of oil, that those resources would rapidly run out. But if you disperse the oil into the water column, it soon dilutes that oil into very low concentration, sort of sub-part per million, quite rapidly, because it spreads it over a much, much wider 3D environment than sitting on the surface, and makes it bioavailable. So with those reduced concentrations, there'll be sufficient nitrogen and phosphorus in the seed to actually, help to actually drive biodegradation. So it really has been an invaluable response strategy for many times. But the response is always different at every spill, and whoever's involved in making decisions at the time, which would include local authorities, depending on which country it's in, but spill response and spill cleanup, and companies themselves will kind of make a decision to use the techniques that will cause the least environmental damage, essentially, and to either remove or have that oil biodegraded as quickly as possible.
Yeah, of course. And obviously the teams on the ground do such a fantastic job in not easy conditions and, quite stressful situations, I imagine, as well. So,
and as I said, particularly on the coast, it can get extremely challenging, because if it gets into sandy and fine sediment, it typically will result in huge amounts of sediment actually having to be removed and dug up and transported away to be remediated elsewhere.
Okay, thank you. Thank you very much. And so, as well as being able to utilise oil degrading bacteria for bioremediation, I did read in your work that oil degrading bacteria do produce biosurfactants. Can you explain why that's important and some of the uses maybe for biosurfactants?
Yeah, so I mean, they're important for the bacteria because oil is a hydrophobic substance. It has very low solubility, and essentially the bacteria producing a biosurfactant is kind of doing the same thing as what I just talked about with chemical dispersant. They're actually making the oil, which is their carbon and energy source, more bioavailable for themselves. So a biosurfactant can be just as powerful as a chemical surfactant in actually reducing the interfacial tension between oil and water, and essentially emulsifying oil into oil and water emulsion to make it bioavailable to themselves. So they put energy into producing these compounds when they're presented with oil, and it allows them to uptake that across membranes into the cell to actually biodegrade. But surfactants themselves have been really important chemicals throughout history, and there's interest in potentially using oil degrading bacteria to produce biosurfactants at scale for industrial purposes. So one thought is that they could potentially be used to replace the chemical surfactants that we find in oil spill. And the benefits would really be that they're a greener alternative, a natural product, more readily biodegraded, because if you're adding another chemical to the environment, that itself needs to be biodegraded as well. And generally, they'd have lower toxicity and environmental impact.
Yeah, the fact that they are so much greener means that surfactant market is potentially huge. the demand currently for biosurfactants really is rapidly growing. It's been valued at around about $1.5 billion recently, so it really is a huge amount. And as well as for being used as dispersants, as we've discussed, they can also be used in a wide range of other more environmentally friendly products, so things like cleaning and personal care products.
Oh, fantastic.
So there is just like a lot of potential for them.
Oh, amazing, thank you. So have you come across any particular challenges with engineering biosurfactant pathways in your work?
Yeah, there are some big challenges in there. So some of the major challenges include understanding the actual different genetic pathways for their production. There are many different bacteria that produce these surfactants, and they produce different types of surfactants. And that means that the pathways are different in basically every bacteria. So like only a very, very small amount of these pathways are currently known and understood. So it is one area that we're really keen to investigate whilst we're working with EBIC, and that we can find a much wider range of these pathways to get a wider range of surfactants that can potentially be produced. And another massive hurdle is going to be actually engineering these bacteria to produce high yields needed, because that is currently something that is just not done very much. So there's a lot of work that needs to go into that. And the extraction processes, the purifying processes, just generally producing them really at a large scale to be used in an industrial way.
Yeah, as Hannah was saying, it is really a kind of an interesting potential growth area, and interesting biosurfactants have been growing. Out of the many types of bacteria, particularly in the marine environment, that can degrade oil, we only actually know the pathways for quite a few of them. Pseudomonas bacteria, for example, produce rhamnolipids. Bacillus type bacteria can produce a biosurfactant called surfactin, and there are some others such as Trihalose lipid, but there's a really wide range of different types of surfactants. And evidence actually shows that a mix of surfactants may well encourage a marine community to degrade oil even better, because they all produce many different types. And some of the organisms we work with that are really specialist hydrocarbon degraders, such as Alcanoborax, which was actually the first obligate hydrocarbon degrader to have its genome sequenced back in about 2006, which was actually led by Peter Gleishin, who is at Bangor and actually works within EBIC as well. Recently, a team in Bonn in Germany, actually characterised the genetic pathway for the production of the sort of glucose lipid surfactant that is produced by Alcanoborax. But we have a wide range of other species where we don't actually know what the genes are. So current databases for bacterial surfactant production are actually quite limited, actually.
Oh, okay, thank you. So what kinds of engineered microbes are currently being explored to break down hydrocarbons more efficiently? How do you determine the effectiveness of oil metabolizing microorganisms? What's the system there?
So we're currently exploring a number of natural marine hydrocarbons degrading bacteria, which includes Alcanoborax, as was mentioned. And all of these bacteria have different capabilities to break down different types of hydrocarbons, because you get some, I not want to go too sciencey here, but get sort of ones that have got straight chain things, ones that have got like aromatic rings on them, and you need different mechanisms in order to break these down. So each bacteria seems to specialise in its own way of doing that.
Yeah, I think you kind of covered the main points there. And I mean, one of the ways that we can kind of determine the effectiveness is, we work with bacteria individually, but also as in communities, that we can look at growth rates of bacteria over, different ranges of environmental conditions. We can measure biodegradation rates by actually quantifying the removal of hydrocarbons. We do that in the lab with a technique called gas chromatography mass spectrometry, which allows us to, accurately quantify concentrations of many hundreds of different hydrocarbons in different environmental matrices. But you know, we're also interested in looking at the genes and particular enzymes that are used in bacteria, because, as Hannah mentioned, because the structure's all different across many different types of hydrocarbons, we find that also the enzymes to actually tackle them and biodegrade them, and particularly oxygenase enzymes, which are a first step in the biodegradation. Hydrocarbons are really kind of inert compounds. They're not very reactive chemically. So the specialist bacteria that can degrade them will actually activate the hydrocarbon by inserting oxygen into it using different types of oxygenase enzymes. And we find many different types. And one of the organisms we're working with has 7 or 8 different types in its genome itself. And that's some of the work we're interested in doing, is currently characterizing exactly what each enzyme can do in terms of degradation of a different range of individual hydrocarbon substrates.
Amazing. So to engineer these microorganisms, how do you go about identifying the genes involved in degradation? What's the process like for that?
So this can include a range of different methods. First, if we genome sequence the DNA of hydrocarbon degrading bacteria, it can be possible to identify some potential candidates simply through computational methods by comparing the similarity of the gene sequences that we find to those that we already know about.
Okay.
Yeah, and that's actually something that Monica, who's part of our team here at Essex as well, has been doing some great work on recently. So she's been isolating a wide range of different bacteria on a whole host of different hydrocarbons, and she's actually in the process of starting some screening processes to see, you know, would they be good candidates to investigate their biodegradation and biosurfactant producing pathways. So, as I was saying, there's a very sort of wide range of different hydrocarbon degradation genes. So we employ a range of different molecular methods to actually try and understand what they are. Some of those that we've used really successfully in the past have been proteomics. So we can grow bacteria on any hydrocarbon of interest, for example, in comparison to a non-hydrocarbon control growth substrate, and simultaneously extract all the protein from the bacteria whilst it's in exponential growth, and essentially quantify and sequence peptides from proteins that we then build back up in against computational databases of the genome to see exactly which proteins are being upregulated and expressed on very different and specific individual hydrocarbons. And we can also do that at the RNA level as well. So Hannah at the moment has just been working on a marine hydrocarbon degrader that specialises in polyaromatics. Monica's been growing this up really successfully and measuring its growth on a wide range of sort of 2, 3, 4 and 5 ring aromatic hydrocarbons, which are really problematic compounds in the environment because they're toxic and carcinogenic. And we're currently in the process of looking at both transcript gene expression and protein expression in this organism.
Amazing, thank you. So from there, how are you engineering the hydrocarbon biodegradation pathways?
Yeah, so one of the things we're really kind of doing in this early stage of EBIC, before we get to sort of building pathways up, or actually, you know, perhaps enhancing or changing them, we're using synthetic biology techniques, engineering biology techniques to actually determine exactly what individual genes and proteins do actually do. And we're using a combination of methods for that.
So I've been using some of my previous knowledge from my PhD work to overexpress identified candidate proteins in hydrocarbon degradation pathways. So we do that in E. coli, that we've basically forced them to make lots and lots of the protein of interest. And then we use that to determine what individual hydrocarbons they can act upon by carrying out enzyme activity assays. And we also try to identify how they're carrying out this activity on a structural level. And I tend to do that using techniques like X-ray crystallography. And so that's a technique that allows you to see things in really fine detail, such as like which parts of the protein the hydrocarbons actually bind to. So you get really detailed structured information from that. And then we can combine that together with the assays to have a full understanding of how these proteins are actually working. And along with that, we're also looking at creating gene knockouts. And so this is where we can remove individual hydrocarbon degrading genes and basically see if that stops the bacteria from being able to actually grow on the hydrocarbons that it would normally grow on. And this helps to confirm the exact function of the very wide range of genes across the bacteria that we're looking at.
Oh, I see, okay.
Yeah, and that's a really interesting area of work that we're currently working on with David Lee Smith's team as part of EBIC, who are based at UEA, and with Amanda and Harry, who are part of his team. You know, they've been creating a number of mutants, knocking out many different individual genes and combinations of genes that we're going to continue some work with shortly to try and confirm that function. Ultimately, further down the line, you know, when we've got a full understanding of all the genes in different steps of biodegradation pathways, you know, we may be able to completely build up hydrocarbon degrading pathways, expand the range of hydrocarbons that particular species could grow on by inserting new function into them, or potentially even insert novel function into faster growing species that could, you know, potentially be used in bioreactors for remediating polluted material ex situ, for example.
Oh wow, that sounds amazing. Really exciting prospects there as well for the future. Is there a way to utilise microorganisms such as the bacteria in oil spill monitoring, sort of like post-spill, for example? Is that something you're also working on?
So yes, that is absolutely possible. I'm actually going to defer to Boyd on this question for the rest of it, because he has actually recently published some new work on this area from an oil spill in the UK.
Yeah, I mean, that's something we've actually been working on for a number of years, as have many others across the world. But, you know, there's a lot more interest in post-spill monitoring of oil, in just measuring the carbons and looking at measuring hydrocarbons over time. And, of course, you know, monitoring the effect on the kind of large animals that we spoke about earlier that are affected. And there's a lot of interest in actually, as part of you know, official monitoring programs, looking at microbial communities and their change over time following a spill, but using that information to actually tell you something about the environment's capability of like remediating the oil from that spill. So we've done this on a number of spills, but what Hannah was just talking about was some work that was led by one of my PhD students who's recently graduated, Jake Smallbone. It was an oil spill down in Poole Harbour in the UK. So it was in the news, obviously, because it was a pipeline spill in the region. But it's if you're not familiar with the region, it's kind of an area of natural beauty, but it's a site of special scientific interest. There's large RSPB bird reserves in the area, and national heritage and protected parts of the environment, and even seahorse colonies that are outside the main harbour, which one of our other PhD students is actually working on, Russ Connolly, which down in that environment. So it's a really precious environment. So there was concern initially where there was spilled oil. I mean, what I would say is actually that oil spill didn't turn out to be particularly damaging, because spill response industry were there very quickly, and most of the oil was contained to a very localized area of the spill site. But we did some work where we were looking at the microbial communities, and what it showed, I mean, coupled with chemical data that had been measured as part of the monitoring campaign, for example, by CFAS, which showed that there weren't really oil hydrocarbons in the wider harbour area. we started to look at total microbial communities and found that they'd only really been altered in the very local vicinity of the oil spill, and we sampled many different areas up the creek that the oil spill was spilled in the wider harbour, and in some kind of control areas where we knew from aerial surveillance that there had been known oil. And what we actually do is extract environmental DNA from the environment, so we can take it from seawater or contaminated sediments. We can sequence and quantify the effects on microbial communities. So using modern techniques such as next generation sequencing, we'll essentially get millions to billions of DNA sequences to assess communities in a wide range of sort of replicate samples across multiple sites. And it helps us to kind of identify if there's negative effects of the oil on microbial communities that form a wide range of really important functions. But it also importantly shows us positive changes in the community, because we can then quantify significant increases in hydrocarbon degrading bacteria. So we can see how they naturally bioremediate in any oil that's not been removed by physical remediation. You know, it helps us to fully show the structure of communities, but also the function, because we look at metagenomics, which is sequencing all the DNA, so we can see exactly what type of genes are present in the environment, and quantifying the increase in hydrocarbon degradation genes. So within that, we can see genes that increase for alkane degradation, which are linear hydrocarbons in oil. We can see genes that we know target polyaromatic degradation. And we can also see whether they're involved in aerobic pathways for the bacteria that live at the surface in an oxygenated environment, and also deeper in the sediments where it's quite often anaerobic processes because of the absence of oxygen. And if we monitor these changes over time, you know, we can actually see when the environment returns to its former state prior to the spill, and also comparing them to unoiled controlled environments at the time of the spill. And we've done this in other spills as well, such as a Greek spill in the Athens Riviera some years ago that really highlighted actually how some oil degrading bacteria, you know, do remain in the environment many months after all the oil has actually been removed and biodegraded, particularly as many of them can pack on storage compounds during the process of oil biodegradation, and incorporate things like polyhydroxyalkanoates into the cell, which they can use as carbon reserves later when the oil has disappeared. But yeah, there's lots of ways that it can tell us important information of how spills are being remediated, and also how successful any response strategies that have been applied have been.
Oh, that's absolutely fantastic. That sounds like really interesting work as well. We'll have to see if we can share a link for your recently published work as well, actually include that with this.
Yeah, that would be great. Thanks.
Yeah, we'll do that, definitely. So before we do finish, I do have some fun sort of quick fire questions for you. Nothing to take it too seriously, don't worry. But first off, just can you tell us a fun fact about yourself, something we won't know?
Shall I start?
Yeah, go on, you can go first.
So I'm going to talk about my hair, which seems like a weird thing. But it does actually relate. So people who know me would know that I have bright coloured hair. Obviously, unfortunately, no one can see that, so they can only hear my voice. But I guarantee that it is a bright, like pink colour at the moment. But the colour actually quite often has been dictated by my work, because a lot of the work that I do involves brightly coloured things. So some of the proteins that I've been working with, because I deal with metalloproteins, they have a lot of colour to them. So for example, one of them that I'm working with in this project is actually bright yellow. It's like a highlighter yellow colour.
Oh wow, I love that.
So I have a habit of, when I find something that's really fun coloured in my work, of taking pictures and sending them to my hairdresser and being like, I want this colour.
That's so good. Okay, we'll have to include a selfie as well when we post this on our socials.
And
Boyd, what about you?
Yeah, well, unfortunately, I can't say the same thing because I don't have much of my hair left, unfortunately. So, yeah, I was thinking long and hard about what might be a fun fact about myself. But what I thought I'd mention is something new that I've got into, which may not sound fun to everyone. But I recently decided that I wanted to get an allotment and kind of go into growing my own fruit and vegetables. And I've only had it for a year, and it's something that I've become a bit obsessed about.
Oh, I love that.
I think it kind of links to my passion for sort of lab work and molecular biology, which you need to be quite detailed and methodological about. So I've got a bit scientific with my growing down at the allotment.
Are you perfecting the allotment?
I am trying to, yeah, but I didn't quite do a good enough job because I won the prize for the second best allotment. So I've got to up my game a bit for next year and try and get that first
top prize. Okay, second is very impressive after only a year. Oh, amazing! Thank you. Next question. Okay, if you could collaborate with any scientist, living or dead, who would it be and why?
I would collaborate with Rosalind Franklin. And the reason for that is actually my first sort of love about science. What really sparked my interest in it when I was in school was learning about genetics and DNA.
Yeah.
And obviously she is like the unsung hero for the double helix discovery. And actually that also ties into my more recent work because her discovery was using X-ray crystallography, which is something that I'm very passionate about using as a technique.
Oh, that's lovely. Thank you. And how about you, Boyd?
Yeah, I'd probably say Craig Ventnor, seeing as I'm a molecular biologist, and it was also partly to do with the fact that when I was a PhD student, I went to my first international conference some 25 years ago, and he was actually the keynote speaker, and he's by far one of the most influential molecular biologists of our time. At that time, he led the sequencing of the human genome on a large project where, from memory, it cost about $4 billion to sequence a single human genome, and that's now something we can do in the lab for a few dollars in a rapid amount of time rather than a few years. The technology has moved on.
So
but yeah, some of the work that he's been involved with has just kept my interest for many years. So moving on from that, they sequenced the genome for many other higher organisms. But he was also, as a marine biologist, led the first kind of really large scale ocean metagenomic sequencing that was discovering DNA sequences that had never been seen in science before from massively parallel sequencing on large projects. And actually the project where they actually engineered the first fully synthetic bacterial genome, so built the whole genomic sequence from DNA synthesis and named the cell Cynthia because it was synthetic. But yeah.
Amazing. Oh, fantastic. Those are really good choices, and I can see why you both picked your respective options there. The final question for you both. If your research had a mascot, what would it be?
Yeah, this is a tough question. I've been trying to think of interesting bacteria, but I kind of went with something else to maybe keep my wife happy. So I thought it should maybe be our mini poodle Rebel, partly because his name is part of the University of Essex motto, which is be a rebel. And he's actually named after the university sports teams who are the rebels.
Oh, amazing.
The Be a Rebel theme is all about embracing the history and activism and independent spirit of Essex. So yeah, we're just not allowed dogs in the department though, so I can't bring him out my mascot.
Oh, bless him. Well, I'm sure he likes to be a mascot from the sofa at home. Yeah. And what about you, Hannah? What would you pick?
Well, mine probably isn't as cute as that because I have to say, having seen pictures of Rebel, it's very cute dog. But I'd probably bring it back to the science side a bit. So you can get like sort of plush toys of bacteria. And what I would love to do is make one of Alcanivorax, since it's like one of the best known oil degrading bacteria, and maybe have it like eating a bit of oil. And I think that would be kind of adorable. A dog's definitely going to beat that.
To be fair, with this audience, it's going to probably equal love dogs and.
Yeah.
I just need to find someone who can make the toy for me now.
Yeah, okay.
I think you could probably do it yourself because Hannah is quite handy in the lab. But when I walked in there yesterday, she was actually making a Christmas tree out of green centrifuge tubes. So yeah.
Oh, brilliant. Okay. Okay, that might be the challenge for our conference next year, Hannah.
Okay.
Yeah. Honestly, thank you both so much for your time, though. I've really enjoyed speaking to you both, and obviously learning more about your research and all the important work that you're doing. Boyd, actually, you're also hopefully joining us for a live webinar on 16th of March next year, which is looking at biosurfactants for oil spill response. Obviously, we'd love for you listeners to join us. If you head to our website, you can actually sign up for that now. We're also really keen to have questions from the audience, so you can send those over in advance if you like from our social channels, or email me, which is info@ebiccentre.co.uk. Our most recent newsletter will actually also be out shortly. So if you're not currently subscribed, do make sure that you head over to the website and join us. It would be great to have you as part of our network. Thank you very much for listening. And again, thank you so much, Boyd and Hannah, for being here today.